Hello everyone and welcome back to Biology with Mrs. Evans. Today we are starting Unit 6, which is on nucleic acids, and this video is primarily going to focus on the scientists that contributed to our knowledge about DNA. And we're going to start our conversation with Frederick Griffith. Frederick Griffith was a scientist who basically discovered transformation as a result of his experiments. So he worked with two types of bacteria. One called the R-type bacteria, and that's because it has a rough coating. And this particular type of bacteria does not cause pneumonia, whereas the S-type, and it's called S-type because it has a smooth coating, did cause pneumonia. And basically what he did was he grew each type on a petri dish, and then he would take each type and inject it into different mice, and he would see if they developed an illness. And the ones that were injected with the R-type did not develop pneumonia, but the ones that were injected with the S-type did develop pneumonia and eventually would die. And what he did was he would heat the S-type and basically kill the bacteria, but he would expose the R-type to that killed bacteria, and what happened is that the R-type could turn into basically the disease-causing bacteria. So he came up with the idea of transformation, and transformation is simply that one type of bacteria can turn into another. So this outlines his basically experiment in a nutshell. He injected the smooth bacteria into the mice and they died because they developed pneumonia. He injected the rough bacteria into the mice and they lived because they didn't develop pneumonia. And when the smooth bacteria basically was killed but placed with the rough bacteria could turn into basically the disease-causing type of bacteria, and that led to, again, the idea of transformation. And this just outlines basically his experiment. So he basically killed the S strain and exposed that basically those killed cells to the R strain, and he ejected that mixture into the mice, and basically they developed pneumonia and died. So he came up with, again, the idea of transformation, that one strand of bacteria can permanently change into another. He didn't know exactly what caused it to change, he just knew that it could happen. As a result of basically his experiments, other scientists set out to try to figure out what exactly was it that caused transformation. The first scientist basically to tackle that was Oswald Avery, and basically what he did is he used enzymes. And the way that I remember Avery and what he did is that, remember, the ending for enzymes is ASE. Avery's last name starts with an A, so that's how I keep it straight, is he used enzymes. So what he did was he used basically different enzymes to break apart the different types of macromolecules. Remember, enzymes are substrate specific, so he would use one enzyme that would kill all of the carbohydrates. He used another enzyme that would kill all of basically the lipids, and then another enzyme that would kill the protein, and then another enzyme that would kill all of the nucleic acids. And what he found is that transformation only occurred when DNA basically was not destroyed. When DNA was destroyed by the enzyme that basically broke apart DNA, transformation did not occur. And this slide here basically summarizes his results. So when lipids were destroyed, transformation continued. When carbohydrates were destroyed, again, transformation was present. When proteins were destroyed, transformation still occurred. When RNA, remember there are two types of nucleic acids, RNA and DNA. When RNA was destroyed, transformation still occurred. But when DNA was destroyed, transformation did not occur. So he basically concluded that the agent, what caused or allowed for transformation to occur, was DNA. And this basically outlines his experiment. So he used RNA ACE, which basically killed RNA. Protease, again, destroys protein. DNase destroys DNA. Lipase obviously destroys lipids. And then carbase destroys carbohydrates. And this is basically the enzymes that he used. And again, the outcome is that transformation stopped when DNA was destroyed, but transformation was still present when all of the other types of macromolecules were destroyed. So he concluded that DNA was basically the agent for transformation. We know that scientists like to look at other people's work and try to come up with an experiment that will basically put it to rest. And Martha Chase and Alfred Hershey basically looked at what Avery did, and they wanted to finally put it to bed, because we know that DNA is found inside the cell, and at that time they also thought that maybe protein played a role as well. So what they did is that Hershey and Chase used what we call radioactive isotopes, and they labeled one isotope was specifically geared towards marking the DNA in the cell, and then another isotope was used to mark the protein in the cell. And then what they did was that they allowed the bacteria to grow, and then they looked for those radioactive markers in the bacteria cells. And what they discovered is that the radioactive isotope that was used to mark DNA was the only one that showed up in the new cells. So they basically put it to rest once and for all and said that DNA was the cause of transformation. And the organism that they used to help them basically was what we call a bacteriophage. A bacteriophage is a virus that is going to infect a bacteria cell. So again, sulfur was used to mark the protein, and then phosphorus was used to mark the DNA. And what they noticed is that only the DNA isotope showed up after basically the cells grew. So once and for all, as a result of Hershey and Chase's experiment, they confirmed that DNA was the agent that caused transformation. So what is DNA? We know that DNA is a type of nucleic acid, and we know from Unit 2 that nucleic acids, their monomer is a nucleotide. And I told you in Unit 2 that we would get into more detail about what a nucleotide is. A nucleotide consists of 3 parts. And some of these should look familiar because we've talked about the phosphate group before. We've also talked about this pentose sugar that you see, and then we've also talked about adenine when we talked about ATP. So a nucleotide consists of a phosphate group, a sugar, and it depends on whether or not it's DNA or RNA, and then the nitrogenous base. And when we did Unit 2, you guys did a hands-on activity and you got to see the different sugars that are in nucleic acids. You know that in DNA, it's called DNA because it has deoxyribose as its sugar, and then RNA is called RNA because it has ribose as its sugar. And remember, deoxyribose means that one of the oxygens have been removed. And then the abbreviations, remember, for the phosphate, we typically draw that as a circle with a P, and then because the sugar is a pentose sugar, meaning it has 5 carbons, we draw it as a pentagon, and then attached to the sugar is the nitrogenous base, and that can be basically however you would want to draw that. You could draw it as a rectangle or whatever representation. So remember that the parts of the nucleotide are phosphate, and attached to that phosphate is the sugar, whether it's ribose or deoxyribose, depends on what type of nucleic acid, and then attached to the sugar is the nitrogenous base. So if you look at this slide, you'll notice that the two of the nitrogenous bases, you have adenine and thymine. We already talked about those four bases when you guys did the hands-on lab in Unit 2. So if you notice, based on this picture, notice how they link up based on shape. So remember, thymine and cytosine are pyrimidines, which means they are basically one carbon rings, whereas guanine and adenine are purines, which means they are two carbon rings. And it's because of those shapes as to how they match up. And remember, we know that adenine always pairs with thymine and cytosine always pairs with guanine. So the four bases, just as a quick review, because we already talked about it in Unit 2, are adenine, thymine, cytosine, and guanine, and we typically abbreviate those as just A, T, G, and C. And remember, adenine and guanine are your double carbon rings. As you see here, there's two carbons here. And then cytosine and thymine are your pyrimidines, which are only one carbon, basically circles there. And again, adenine bonds with thymine, you can see how they match up, and then cytosine and guanine match up. And again, you practice that in Unit 2 on the hands-on activity that we did. So that brings us to Erwin Chargaff, and Chargaff was a scientist that basically did an assay. An assay basically would be a survey. He would look at basically the percentages of adenine and thymine and percentages of cytosine and guanine in a sample of DNA. And what he found is that each time he ran the assay, adenine always was the same percentage as thymine, and cytosine was always the same percentage as guanine. And that led to the idea that adenine always paired with thymine and cytosine always paired with guanine. And this is known as Chargaff's rule, based on his findings. And we also call it the base pair rule, because we know that adenine is going to bond with thymine and cytosine is also going to bond with guanine. So because of his discovery, we know that we can calculate the percentages of adenine based on thymine, and then we can also calculate the other two percentages, because remember, it's always going to be out of 100%. So for example, let's say that you know the percentage of adenine. It should equal, in the example here, it's 30%. So if adenine's 30%, thymine must be 30%. You add those two together, it gives you 60%. Subtract that from 100, you're left with 40. And divide that by 2, because you still have two bases left. So that means cytosine is equal to 20% and guanine is also equal to 20%, because again, they must be equal. So here's an example. You have 20% adenine. So you try to figure out what the percentage of thymine is and also what cytosine is and guanine. You might want to pause the video and take a moment to calculate that. Alright, so adenine is 20%, so we know based on Chargaff's rule that adenine pairs with thymine. So if adenine's 20%, then that means thymine is also 20%. So you add those two together, you get 40%. Subtract 40 from 100, you're left with 60. And that means that 60% of the survey was cytosine and guanine. So you divide that equally, because we know that they must be equal. So that means that cytosine is 30% and guanine therefore is also 30%. So that's how you would calculate that, and you'll need to be able to do those type of calculations on a quiz, test, etc. Double helix. So you guys have all told me when I asked you what the shape of our DNA is, you guys have said that it was a double helix, and it was because of the work of basically three scientists that came up with basically that shape. Rosalind Franklin and Maurice Wilkins are the scientists that used X-ray diffraction, and basically for me and for you as well, that just simply means that they took a picture of DNA, and what they came up with is what you see here on the screen. So to you and I, it looks like basically an X on a film, but to Watson and Crick, who had spent many years trying to figure out the shape and structure of DNA, it finally clicked for them that DNA was in fact a double helix. So basically what Watson and Crick did was they took the work of Chargaff and they said, okay, Chargaff said that adenine always pairs with thymine and cytosine always pairs with guanine, and they came up with the double helix based on the picture that Rosalind Franklin basically took of DNA. So it's because of Watson and Crick that we came up with a model of DNA, and again, you know it as the double helix. So using, again, the work of Chargaff and Franklin, Watson and Crick came up with a double helix. We know that the sugars and the phosphate, so for DNA it would be deoxyribose, those are what we call the backbone of DNA. So they're on the outside. Those are basically the sides, if you will, of the ladder. And then the rungs on the ladder, those are your nitrogenous bases. So adenine and thymine, cytosine and guanine, those are the basically the steps on the ladder. And again, Watson and Crick came up finally with the model, and they said that it was a double helix. They had been working for many years to try to figure out what it was, and it wasn't until Franklin's picture that she basically published that they had the aha moment that, oh, it's a basically a spiral staircase known as a double helix. So DNA model, this is something that you will need to be able to basically describe what's going on. So again, the phosphate, notice here, and the sugar are what's going to form basically the sides or the backbone of the DNA molecule. And then the steps on the ladder, the inside, is going to be your nitrogenous bases. So you can expect to see a picture like this, and you'll be asked to identify basically the different parts. So X that you see here on the screen, again, this would be a nucleotide. Here's the sugar, here's the phosphate, and then here is a nitrogenous base, the three parts of a nitrogenous, of a nucleotide. And then the circle, we know always we use a circle to represent phosphate, and then the pentagon, remember I said pentagon is usually used to represent the pentose sugars, so this would be deoxyribose, and the three parts makes up a nucleotide. This guy right here, cytosine, adenine, guanine, those are your nitrogenous bases, and what they're showing right here, this C and G, is what we refer to as a base pair, because they are basically bonded together. And that brings us to basically the structure of DNA. So the backbone of DNA is the sugar and phosphate. Those two are bonded together with a covalent bond. And remember from Unit 2, those are our strongest bonds. And then adenine here and thymine, these two are going to be bonded together with hydrogen bonds. And remember, hydrogen bond is the weakest bond. And it's a weak bond between the two because the cell is going to need to split basically the two strands of DNA in order to copy DNA. So because this is a weak bond, it's easy to split basically the two strands apart. And again, you're going to want very strong bonds on the side because you don't want those to break apart. Because if they broke apart, then it would be very difficult to copy the DNA. So you have covalent bonds here that form the backbone between the phosphate and the sugar, and then you have hydrogen bonds that form between the base pairs or the nitrogenous bases. And again, it's a weak bond so that it can easily be split apart to form two strands that will be used to make copies of DNA. So DNA replication, a replica basically is an exact copy of something. So DNA replication happens when you have several enzymes that are involved, and we're going to go into more detail with this in the next video or so. But basically what happens, again, hydrogen bonds are going to be broken by an enzyme called helicase. And that is because, remember, the shape of DNA is a double helix, so it's going to split apart the DNA molecule. And what happens is you have your two strands that are exposed. Another enzyme is going to read those exposed enzymes, and they are going to bring over the matching base that would bond with that. So as you see here, this base would be adenine, so it's going to bring over a thymine to pair up to it. Again, this is adenine, so it's going to bring over a thymine to pair up to it. And the enzyme that does that is called DNA polymerase, and we will talk about how DNA replication takes place further in the videos to come. So for this video, know the scientists and their contributions to what we know about DNA, know the parts that make up a nucleotide, know how they bond together. So nitrogenous bases bond together with hydrogen bonds, and then the phosphate and the sugar, which are the backbone, are bonded together with covalent bonds. So one thing about DNA replication, we would say that the two strands are complementary, and that is because one strand of DNA can code for the other strand of DNA. So here on the screen you have a DNA sequence is A, T, C, G, C, T, T, C, A. So how DNA polymerase works is it is going to read that strand, and then it would bring over the matching bases. And the way that you would do that is using the base pair rule. So A always bonds with T, T bonds with A, C bonds with G, G bonds with C, again C bonds with G, T bonds with A, T bonds with A, C bonds with G, A bonds with T. So you can use one strand of DNA to make the complementary strand. And basically I showed you how DNA polymerase works. It reads the exposed DNA, and it would bring over the matching strand, as I have written here on basically the screen. And that's DNA replication. Quick look at it, and we will again go into more detail in the next video. So those are the basics for this video that you need to know. Thanks. Have a great day. See ya. Bye.